Good evening, ma'am.
Good evening, sir.
Good evening.
Sir, you can start. Sir, are you there?
Hello, ma'am.
You can start, sir.
Okay, ma'am. Thank you. Thank you so much. Good evening, my dear students.
Good evening, sir.
Good evening, then listening. Okay. Last class, what are the things already we discussed in biology classes? Already you know, isn't it, my dear? So, what are the types of what is meant by PCR? What are the components of PCR already we studied? First of all, template and primers, and then what are the properties of primers and energy requiring process. Suppose if it is energy required, that is denoted as the deoxynucleotide triphosphate. You know, remember DNA containing 4 nitrogen bases, the 4 nitrogens also included. Okay, whether it may be adenine, thymine, guanine, and cytosine means adenine deoxynucleotide triphosphate. thymine, deoxynucleotide triphosphate, guanine, deoxynucleotide triphosphate, and last one is called as cytosine. Okay, not only that, and also that divalent cations. Already we discussed. There are two things is very, very important for their enzymatic activities. One is called as a magnesium, is also already known. And manganese, Mg and Mn, both are divalent cations. Okay, they, according to this involvement of these components, the cofactors, what happens? It enhances the rate of reaction. That is very, very important, my dear. Okay, enhance the rate of reaction. That means speed of the reaction. Rapid reactions also takes place by the things also I will be discussed. And the next one is the thermal cycles. Thermal cycles is nothing but the machines carried out by PCR. Okay, the PCR is also carried out by the one of the specialized equipment is called as, what is that, a thermal cycler. Okay, it's also work based on the principle that is also very, very important. It is also, it has a specific principle also. That principle is called as, what is it, Peltier or Peltier effect. Okay, that is very, very important. Then only the process will be completed. Okay, that is also very, very important one. Next, we are going to discuss how it will be. Okay, what are the primers, what are the DNA molecule, magnesium ions, as well as the instruments, how it will be act. That things we are going to discuss in stepwise. What are the procedure? Okay, what are the procedure under the PCR technique? That things we are going to discuss. First of all, melting of target DNA denaturation. First of all, you should note it down. You have to do the first step, which one is melting of target DNA. Otherwise called as denaturation. You have to isolate the DNA or melting target DNA. Which one we are going to amplify? The DNA was isolated from the different sample. Okay, already we discussed that. Then that things also, whether it may be a plant or animals. Sometimes a plant means a whole part of the body. Already in our lab, particularly in our lab, the plasmids. Okay. And suppose if it is a somatic hybridization and monoclonal means, what is that? A tissue culture is also very, very important. Okay, that is plant samples. Animal means already in our lab. Okay, animals also, it has a specific structures. The mitochondria also, it has a specific structure of the plasmids, and also animal cells. And take an example of WBC. That is very, very important. Nucleic acid present in it. It can be denatured and identify the particular molecule and also isolate, and then it undergoes the denaturation process. Okay, that is the first one. Okay, the melting of target DNA denaturations, the target DNA containing sequences between 100 and 500 base pair. It should be how many base pair? It should be present, 100 to 5,000 base pair to be amplified is denatured at around 94 degrees Celsius for 20 to 30 seconds. When you are giving the temperature up to this level, already in our lab. Okay, what is by denaturation? DNA denaturation already in our lab, isn't it? How it will be possible? Take an examples. Okay, take an examples. This is considered a double standard DNA molecules. Double standard DNA, isn't it? So double standard. So DNA molecule. Just imagine this is a structure of double standard. Okay. Okay, see this one. So this is considered here. Okay, double standard DNA molecule. Okay, how it will be denatured? How it will be get? So likewise, how it will be get it? Okay. So in between already in our, there are so many nitrogen bases in between. Okay. Already it is also connected with the nitrogen bases. Nitrogen bases in between is there. Okay. There are so many nitrogen bases also there. Adenine, thymine, guanine, cytosine correspondingly, it matched with the help of the bond. Already we know. This double-handed helical structure, how it will be splitted. Okay. In between there is a hydrogen bond is there already in our board. Adenine molecule combined with the molecules of, which one, adenine always combined with the molecules of Thymine. Guanine, already you know about it. It's always joined with the cytosine. Okay, in between there is a bond. Is that this bond is called as what is it? Hydrogen bond. So this is called as what is a 2 hydrogen bond. 2 hydrogen bond. Okay, that is very, very important. Here 3 hydrogen bond is there. Okay, how it will be split? When you are giving the temperature, that is nothing but the heat. That is nothing but the heat. So during this heat temperature, or what happened, what will have the symbol already in chemistry, isn't it? The heat. So the double stranded helical structure in between the nitrogen bonds break at it, and it gives a single strand like this. Denaturation happen inside the molecule. How it will be already but this is the adenine. Okay, so this is the one. So in between guanine, okay, and then cytosine, and also adenine, like that. Okay, this is the first strand. If you take an example, this is considered a 5-diazine, and opposite side already about there is a 3-diazine. So here the 5-diazine, here what happened? This is a 3-diazine. Okay, 3-diazine. Here there is a 5-diazine. After the temperature, what happened? The nitrogen base break out. So break it. So as a result, we will get it the single strand like this, isn't it? Single strand like this. Okay. Take in examples. So this is another part. A always combined with the molecules of thymine. Okay, thymine. Okay, thymine. Guanine already know about cytosine. So here cytosine is there. So guanine comes here. So and also adenine here. The thymine is here. So these are the nitrogen. In between there is a bond. Completely what happened? Broken. Okay. In between. So as a result, what happened? You will get the fragments. Which temperature they are giving? 96. That is already about 96 degrees Celsius. Already we discussed in the TAC polymerase enzyme, TAQ. Okay, TAC. So what is the meaning of TAC? Already about thermus aquaticus. Thermus aquaticus. Thermus is nothing but the heat, isn't it? So this type of microorganism, the cell wall, it contains the enzyme, which one DNA polymerase. So that enzyme, it has a highly activity up to the level of 96 degrees Celsius. Normal enzymatic activity will be how many temperature? 42 degrees Celsius. That is very, very important. This is the normal one. This is the normal one. This is the normal. This is the extreme one. Okay. So as a result, you will get it the 2 single strand like this. Okay, got it? Okay. See that one. So how many minutes it will take? 94 degrees Celsius. Already I told you 96 degrees Celsius, isn't it? Both are similar. For how many seconds it will be essential? 20 to 30 seconds is enough. 20 to 30 seconds. Okay. So the DNA molecule completely denaturation, and also to separate its complementary strand, this process is called as melting of target DNA. First of all, you should select the DNA molecule and split the molecule. Then only, okay, the desired gene it will be obtained. With the help of the heat, how many temperature you have to give, how many base pair it should be there. That is also very, very important. After how many seconds? That 20 to 30 seconds only it's enough for the separation of the molecules, and also melting of the target we can easily isolate it. Okay, that is very, very. Excuse me. This heating to high temperature causes the 2 complementary strands to separate via denaturation or melting. That things already we discussed in the slide. Okay, go to the next one, my dear. Okay, go to the next one. In PCR, the 2 strands of the DNA molecules must be separated in order to allow the primer to anneal to them. What does it mean by annealing? Annealing is nothing but the cooling process. Okay, annealing is nothing but the cooling process. How it will be possible after the separation process? Okay, undergoes the amplification. Number of amplification is also done. After that, what happened? The 2 strands, if you want to obtain, means kept under the low temperature. Kept under the low temperature. That process is called as annealing. This reaction is usually carried out for around 45 seconds. Okay. Denaturation only 30 seconds, but annealing is how many minutes? 45 seconds for about 30 to 40 cycles. That is very, very important. That is very, very important. What is by 40 cycles? See your next slide. You can see it. What is by 40 slides? Okay. I will tell you. So that things also. Okay. See here. So this is the PCR replication process. This is the DNA molecule, isn't it? This is the DNA molecule. After the separation, the DNA molecules also separated single stranded. The primer is added and also leading. Okay. So as a result, the single DNA molecule, it will produce the exact copy of the 2 DNA molecule. Okay. This is the first cycle. When it completed up to this level, 1, it gives 2. That cycle is called as first cycle. After that, what happens? The 2, it gives 4 DNA molecule. So that is the second cycle. So then the third cycle, okay, second cycle, after the second cycle, the result will be 4. Okay. And again, the third cycle, what happened? The 8, it will be produced. Each one it carries 2, because each one is split into 2. Here 2 stand, here 2 stand, here also, and also this one. Exactly, isn't it? Respectively. So it will be split, and also they produce their own, what is that, complement region. As a result, you will get it the 8, isn't it? 2, 4, and 6 and 8. So one copy after 3 cycles, you will get 80 mean. So after 25 cycles, how much it will be? Imagine, you calculate it. So that is also very, very important, my dear. So many times they were asked the question, after the replication process, that 24 cycles or 40 cycles or 50 cycles, it will be finished, means how many number of DNA molecules you will get it, you will obtain. Likewise, they ask the questions. I will discuss then that things in later. Okay. So, okay, see. Okay. So the 40 to 30 cycle, 45 seconds, how many cycles it will be finished there? So for 30 to 40 cycles per second, one cycle will be finished. So see the speed of the reaction. Okay. So as a result, take an example. 45 seconds means you will get 30 to 40 cycles. Suppose take an example, say 45 minutes means 4.5 minutes means how much is it? This is also multiply into 10. So 400 cycles you will get it. So 45 minutes means you can multiply into 100 again. So how many cycles it will be finished within an hour? That is very, very important, my dear students. So many times it is also asked by the NEET question. See this one. So this is the first step you are doing. This is the double stranded DNA molecules already in overall board. Under the first step, the denaturation process, there 94 is given, here 95 is given. Okay. So exactly 94 to 96, 94, 95, 96, whatever, 95 exactly. Okay. 95. So after that, what happened? The 2 double stranded helical structure in between the nitrogen bases split. You will get it the 2 strand like this. After that, we are adding the DNA primer. Okay, DNA primer already in our about. How many nitrogen bases also present in it already we know about. When you are adding the primer, okay, this is called as a reverse. This is called as a forward primer. This one is called as reverse primer. Okay, we are adding means it joined with this one. You know, that time the temperature will be reduced up to the level of 50 degrees Celsius, 55 degrees Celsius. 40 degree, yeah, you have to reduce. Then under the primer, it is attached with the complementary gene of the DNA molecule. So this is the another step with the, which one you know, that nucleotides. Already we discussed the DDTP, that is called as, what is that? Thymine pyrophosphate and cytosine pyrophosphate, adenine pyrophosphate, and also guanine pyrophosphate. These are the enzymatic reactions. Enzymes, it's an energy requiring process. Okay. So as a result, the primer is also attached. After that, TAC enzymes also. So it will lead the reaction. This is called as, what is that, you know? Forward. This one is called as backward. As a result, you will obtain the 2 exact copy after 40. Repeat the cycles again and again. Okay, 40 times 4 DNA molecule you will obtain, isn't it? After the second cycle. It will repeat it means what happened, you know? Up to particular durations, you will get it the number of things. Okay. Any doubt in this part, my dear? Dhanalakshmi? Okay. So listen here. This is the DNA molecule splitting with the help of the temperature 95 degrees Celsius. You will obtain 2 single strand. After that, we are adding the primer, DNA primer. The DNA primer should be the particular nitrogen base is also there. Already know about it is also made up of the nitrogen base pair. Okay. Join together. During the joining process, annealing. Annealing means the temperature will be reduced. Okay. Up to 95 to 55 degrees Celsius. After that, the primer is actually exactly complemented with the DNA. Complemented with the DNA molecules. And addition with this, the energy also very, very important one. Energy recurring process. What are the energy? It is also nothing but the, what is that? Thymine thiophosphate and cytosine thiophosphate, adenine thiophosphate, and guanine thiophosphate. These are the essential. Join together. After that, attack. Okay, thermoaquatic acid. Then the reaction it will be proceed forward as well as a backward reaction. Final completion of this step, you will get it 2 copies of the DNA molecule. After 3 to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32, like that, it will be produced after the 40 minutes. How many cycles will be finished? You know, 30. Okay, that is very, very important, my dear. So don't forget, this is very, very important, my dear. See that one? So DNA primer, so here the DNA molecule is split into 2. After that, we are adding the primer, same. Okay, so the forward reaction and backward reaction. This one is called as forward, this one is called as backward reaction. So you will get it the 2. So in this category, which one is the parental DNA? Means the above the mentioned the blue color is the, it is the parental DNA. So here are the lower one is a parental DNA. Suppose in between these two, it's also enter into the another cycles. You will get the exact these two also split, isn't it? So these two are split and these two are split. So in center there is a new DNA molecules also formed. Again and again it repeated means what happened? You know, the process is continuous. The process is continuous. The first stage is called a denaturation, and then annealing process. Adding a primer is called as annealing process. Then elongation. Elongation. What is the meaning of elongation? It's produce a number of copies. Okay. So after the finishing cycles, it will take some more times. See that one. See the next one. Annealing of primer. The second step is the annealing of 2 oligonucleotide primers to the denature of the DNA strand. The primer added in the excess and the temperature is lower to about 50 to 68 degrees Celsius for 35 to 45 seconds. So then the primer it will be easily complementary with the DNA molecules. Okay. Otherwise, what happened? No, the DNA molecule, the primer, it will not connect with the parental DNA. Consequently, the primer forms hydrogen bond that is annealed to the DNA on the side of the DNA sequence. That is very, very here. You must note it down. The temperature is very, very important. Okay. How many minutes you have to put it under the temperature? That is also very, very. These 2 points only. 50 to 68. So here 35 to 45 seconds. That is also very, very important, my dear. So this is important things. The resulting bonds are stable only if the primer and the DNA segments are complementary. This is the important point. Okay, then only the reaction it will be continuous. Suppose if it is not complementary with the primer, what happen? You know, automatically it suffer. Okay, the PCR reaction it will not continue. Okay, go to the next one. The annealing temperature is very critical because if it is very high, primer does not bind to the template and the primer and the template remaining dissociated. Okay, temperature is very, very important for the annealing process. Suppose the temperature it will not under the 55 to 65, 50 to 68 degrees Celsius means what happened? No, it will not exactly complementary with the DNA molecules. It can be dissociated. So these are a human error. Human error we will do during the PCR reaction. Primer is very, very important, in particular temperature very, very important. So then the process will be continue. And if the temperature is too low, non-specific binding occurs. This is also another thing, sir. Annealing already you know about. Suppose the temperature should be 50 to 65 degrees Celsius. Suppose the temperature below the 650 degrees Celsius, when what happen, you know, non-specific binding is also occurs. Okay. It is not proper binding. It is a non-specific binding occurs. As a result, the product will be useless. That is what desired copy it will not, you will not obtain. Okay. It may obtain the different consequences, different sequences. Okay. So that is the important one. You should note it down. That's why it's very accurate. You have to do the accurate manner. Then only the result will be 100%. Get it. Okay. So see the next one. The annealing step is usually carried out at the temperature of 3 to 5 degrees Celsius. The temperature is called as what is that? 3 to 5 degrees Celsius. Lower than what is the TM of the primer. What is the meaning of TM? Melting temperature. The melting temperature should be 3 to 5 degrees Celsius. Okay. That is very, very important. Annealing steps is usually carried out. Just read the question when you are attending the exam. Okay. Sometimes they ask the question, the annealing process which occurs within the temperature, like that they may ask the question. Usually carried out by the temperature, like that they may ask the question. Okay. Melting temperature. Melting temperature, you know. So melting means the first letter M. Temperature means T. That is the actual word or letter, isn't it? Here they have given the reversible. For example, MPBS. What is the expansion of MPBS, my dear student? Will you please explain? What is by MPBS? Hello, Dhanlakshmi. Hello.
Yes, sir.
What is by MPBS?
So, Bachelor of Medicine and Bachelor of Surgery, sir.
Yeah, it is a reversible, isn't it? Reversibly,
Yes,
we are okay. So likewise, you have to read this one also. TM means melting temperature, which is centigrade, you know, Celsius. So 3 to 5 degree Celsius. Okay. So next one is a primer extension. Sometimes they were use the primer extension. First step is finished. Annealing is also finished. Third is okay. Primer extension. How it will be, reaction will be completed. The final nucleotide triphosphate. So these are the triphosphate. Already we discussed this part also. These are the triphosphate. Okay. See, adenine, guanine. Okay. Adenine and guanine, thymine and cytosine. See, adenine triphosphate, guanine triphosphate, cytosine triphosphate, and also thymine triphosphate. These are all very, very important. And yeah, thermostable. That is also very, very important. Thermostable nature. Okay. It will not easily denature. Suppose if it is denature, what happens? No, the reaction is also completed. DNA polymerase are added to the reaction mixture. That is also very, very important. DNA polymerase is also added. Enzyme, this is the enzyme. This is a cofactor. Already the subject is a DNA molecule present in it. So three are involved. Then the DNA polymerases accelerates. Accelerate means the period of reaction. It will be proceed. The polymerization process of primer and therefore extended the primers at 68 degrees Celsius. Okay, resulting in the synthesis of copy of a target DNA sequence. So this is the normal 68 degrees Celsius. First one 96. Then the temperature will be reduced. Then when you are adding the which one, you know, 50 to 60, isn't it? Then you have to annealing process 3 to 5. Then extension process. First one is denaturation, annealing, and extension process. Okay, denaturation already in 96, 94, 95, 96. So here 95 has given. Next, what is that? Annealing process. Annealing process already in our 50 to 65 degrees Celsius, particularly the normal temperature will be 3 to 5 degrees Celsius. It will be very fast in nature. Okay. And then primer extension. It's also very, very important. The temperature is very, very important in this category. How many temperatures? 68 temperature. So these are important process. Suppose it is not exceeded the 60, 70 and above, it is not below the 60 and below. Okay, likewise, you have to calculate. Then only you will get the result. The medium temperature you have to maintain. Okay, 60 to 70. So that is very, very important. Then only the reaction will be elongated. The PCR reaction is elongated. The elongation process also done. Suppose if it is not like that, whether temperature is increased or whether temperature is decreases, it will lead to the false result. See that. In this step, the temperature is again increased in the time to 70 to 75 degrees. The temperature very, very important in this PCR technique. 4 times already we changed the temperature, isn't it? First one, TNIIT. Second one, annealing. Third one, annealing normally which temperature they will do. And then elongation process, what will be. And the last one is increase. This is the optimum working temperature for the polymerase. Okay, polymerase used which add further nucleotide to the developing of DNA strand. This is an additional one. Suppose when we are adding the additional DNA molecule to the PCR technique, we should maintain the temperature up to the level of 70 to 74 degrees Celsius. That is very, very important, my dear. Okay, then each time these 3 steps are repeated, a number of duplicate DNA molecule is doubled. Already we discussed in the diagrammatical aspects. After 20 cycles, note it down, after 20 cycles, approximately 1 million molecules are cloned. How many millions? 1 million molecules are cloned from the single segment of double-stranded DNA. This is the exact result. We should expect. Okay, that is the important one. Short duration. Okay, how many? After 20 cycles, you will get million molecules from a single segment. Single segment means already know any part, 100 or 50 or 200 or like that. There is a specific nucleotide base pair is also there. It should be. Suppose if it is increases, not a problem. It's also lead to some problem. Suppose decreases, it can be very fast. Only those DNA polymerase that are thermostable, that is function at the high temperature, are employed in the PCR technique. That is very, very important. Already told you. PCR technique, which one is very, very important? The temperature is very, very, they play a vital role in the PCR reaction. Okay, it may be changed each and every process. Okay, denaturation, annealing, and also elongation process. And finally, the speed of reaction. When you are adding the additional DNA molecule to the PCR technique, we have to reduce the temperature, maintain the temperature, then only you will get the result. Then the PCR machines can carry out 20 cycles and amplify DNA 105 times in 75 minutes. So this is the main point. It depends upon the instrument also. Okay, sometimes the instrument will be very fast in the PCR machines in higher, that means in America and Japan. Okay, and also other countries, Chinese peoples also, they are having the, the speed is very, very important. Okay, the PCR machines, it has a speed. Okay, here the PCR machines can carry out 25 cycles and amplify DNA 105 times in 75 minutes. The reaction can be continued for 24 to 45 cycles. Okay, this is very, very important. Under normal condition, the amount of TAC DNA polymerase become limiting after 30 cycles of amplification. Normally, the TAC DNA polymerase, how many cycles it will be? No, to carry out, 30 cycles of amplification. This is the limiting factor. This is the important one, and also the limiting cycles also done by the TAC polymerase, DNA polymerase enzyme. This is the important one. Okay, don't forget, my dears. This is a, sometimes they may ask the question, the TAC DNA polymerase become a limiting after 30 cycles. Okay, how many minutes? They will ask the question. How many minutes? How many cycles? Okay, like that they may ask the question. Here already know about 75 minutes, how many cycles it will be finished. Okay, so these are the important points. They all are asked in the mathematical question aspect, mathematical, max oriented. Okay, see the next one already we discussed so many times. Given this is the single strand DNA molecules undergo the denaturation process up to the level of 96 degrees Celsius. You will obtain the two strand. Then it's also a primer added annealing. After the annealing, okay, so undergoes the process that is a elongation process. Here the 3 cycle has given, okay, up to 8. So after 25 cycles, how many you will get, millions of copy already we discussed. Okay, see that one. See that flow velocity. This is the exact term they have given as a tabulation aspect. How many cycles, extension time, cycles time, rotation speed of magnet, RPM rotation per minute, and PCR reactions mixture second -1. Complete time for 25 cycles per minute. Okay, so these are the different things. Extension time 3, 6, 9, 12, 15. This is also threefold, 3, 6, 9 like that. So likewise, 3 means you will get it the cycle 9 times. So it's also double, isn't it? See, according to this one, 9, 18, 27, 36, 45. Rotation, see here, 6.67, 3.31. It will be, what is that? Reduced here. The rotation per minute is reduced in manner. 6.67, 3.37, 3, 2.22, like that. PCR reaction mixtures per mm means 7. Okay, flow velocity. It's also already in our, but the PCR tube is there. The tube is also loaded by the DNA molecule. Already we discussed it. Okay, so 3.49, 2.32, 1.7, 1.4, like that. The velocity will be depends upon the cycles. Complete time for the 25 cycles. Complete time. Okay, suppose if it is 3. Okay, the cycles time is 9. So 6.67 is the rotation speed of the magnet. And then this is the flow velocity. So we'll get 3.75 cycles per minute. So likewise, they calculate 7.5, 11.25, 15, and 18.75. Okay, these are the PCR activities. Okay, so this is the important things, my dear students. Next, we will pass on to the next one, is application of PCR technique. Why we are doing the PCR technique? Why they need? Okay, already we never about amplification of the DNA and the RNA. This is very, very important. First point is very, very important. Okay, suppose if you want to know any one of the DNA molecule, it has a specific character for the very useful one for the social means, or so we have to amplify that things. How will you? Means with the help of the PCR technique only. The next one is diagnosis of disease and causal microorganisms. This is also another important one. Okay, after the amplification, we can identify the DNA molecules and analyze the disease, which disease it will come. You know very well about one of the major concept. You are having 2 meter DNA molecule, isn't it? 2 meters, 3.3 into 10 to the power of 19, isn't it? Base pair sequence is also there. Any particular sequences, they may mention the which age, which disease you will affect. Okay, suppose if it is affect before you known means what happened, you can escape from the disease. Okay, so that is a reason. And then third one is called detection of genetic disease. That is also a similar one, such as sickle cell anemia, phenylketonuria, and also the muscular dystrophy. In the which one, in the embryo stage. Embryo stage, suppose if you identify, because it is a chromosomal recessive gene inheritance diseases, isn't it? So that time, how will you rectify these diseases? Before you should identified which gene is responsible for the sickle cell anemia, phenylketonuria, or muscular dystrophy. That gene, suppose if you find it, I found it, you can remove it from the normal genome. Instead of the infected or disease gene, we can replace it, mean you will get the good quality, good result of the test. Okay, so that is important one. And then fourth one is a 4 in 6 signs. It is also very, very important. Already we discussed DNA fingerprint is also essential for the forensic science, where it is used to identify criminal through the DNA fingerprint technology. Already told you, criminal fingerprint technology. Okay, so this is the another point. And then in diagnosis of the plant disease, not only the animal disease, and some of the plant disease is also done by, already you know about the tumor inducing is also one of the plant disease. BT cotton. These are all are comes under the condition known as, which one you know? Disease due to the DNA molecule, particular sequences. Okay, so we can diagnose the plant diseases also. This is another one. Generation prop. It is an important generation prop. What is the meaning of generation? Your parental character, grandparental character you are having, isn't it? That character identification is very, very important. Okay, we can prepare the prop. Okay, that prop is used as the next generations. Okay, and then generation of the cDNA, complementary DNA libraries. After that, identification of the sequences, data, all those things are stored in the form of cDNA library, complementary DNA libraries. Okay, for future uses. That is also very, very important. Analysis of the mutation. Suppose take an example, A and B. Okay, we are all are C, like that. Imagine A and B is the normal one. We are the mutation means, what is the mutation occurs? How it will be occurs? How will you identify means with the help of the DNA sequences? So which gene is amplification takes place due to the termination? Chromosomal abnormalities already studied, isn't it? Which gene is responsible? Okay, which DNA molecules it may be deleted or addition or substituted, like that we may analyze. Okay, so that is also mutation. What is my mutation? Already studied. The sudden changes occurs. Okay, physical and chemical and biological properties. As a result, the DNA molecules undergoes the changes, sudden changes. Okay, so that is also one of the evolutions. Okay, next one, the same things is given here. To study the evolution using DNA from archaeological sample. This is also important. After RNA copying and mRNA quantitation by the so-called RT-PCR method. Okay, this is the important method. Sometimes they may use the archaeological department. Okay, after the collection of sample from the any one of the animal or plant. Okay, then from that one we have to do the mRNA, identify the mRNA. Then it is also amplifications. The particular DNA molecule is responsible for the growth of the. Suppose take an example. If you want to fly. Okay, suppose if you want to fly, what will you do? You don't have the wings, isn't it? So which gene is responsible for the wings, production of wings? We have to identify that genes. After the identification, we have to isolate. After the isolations, what you have to do now? The same process. What is that? You have to do the PCR method, number of copies. After that, it may be implemented into the fetus. Suppose if you are doing like that, so after the child, you will get the wings. But if you cannot fly, you can try to fly. Okay, so these are the practice. So these are the another things. Okay, go to the next one. In paleontology, what is the meaning of paleontology? Already you know very about it. Okay, PCR used to clone a DNA fragment from the mummified remains of humans and the extinct animals like woolly mammoths and also dinosaurs. Paleontology, the science that deals with study of the fossils. Okay, the woolly mammals and also dinosaurs now completely eradicated from the world. Okay, or globe, whatever it may be. We all are buried under the soil due to the natural disaster. Okay, but just you see that a dinosaur, Jurassic Park in the film already saw it, isn't it? See that one, there is one of the mosquito is there, mosquito inside there is a blood sample is there, that blood sample is containing WBC. The scientists isolate the WBC and the DNA separation, and also they maybe produce a new dinosaur world. Okay, so that is the paleontological studies. And then genetic testing, PCR technique has recently become powerful to detect the disease-associated gene to predicate the presence of heart disease and cancer. This is a milestone of recent technology. Suppose PCR technique has recently become a powerful tool to detect the disease-associated genes. Which gene is responsible for the heart attack? Which gene is responsible for the cancer cells? You can easily identify. So that gene is supposed present in any one of the person, he suffered by the, he will suffer or he suffered. Okay, heart disease or cancer, like that. We can easily identify. So find out the gene and then remove it and remove it. Okay, that is the another important things. Don't forget. And then a PCR is a veterinary parasitology. Veterinary means animals, isn't it? Plants means already in our animals means take an example. All the animals. Owing to the its rapidity and sensitivity as compared to the antibody-based diagnosis, PCR made its use in almost all aspects of biological work, including veterinary clinical diagnosis. Veterinary clinical diagnosis. This is also important. Simple things, mostly used for the animal diseases, identification, isolation, all those things. Next one is very, very important, my dear students. Next one is very, very, very important one. You just note it down when you are going to study the higher classes also. What is my phylogeny analysis? Okay. For example, you are studying so much of microorganism, bacteria, virus, plasmodium, okay, and so much there. Each one it has a specific, what is that? Structure and properties and also functions also, isn't it? But at the same time, what happened? How it will be derived? For your all comes from the your grandparent, your parents or comes from your grandparent, grandparent, likewise, isn't it? So when you are constructing the tree, your grandparent and grandparent, and so your father and mother and you and your generation also, you have to create your tree, isn't it? So we can easily identify which animals is belong to the or related to the. That is very, very important. Okay, there is a link between you and your grandparent by this methodology only. See that the phylogeny of organisms like plant or animals and other lower organisms can be traced out by DNA analysis. Okay, the origin of unknown sample like the recovered bones of early men can also be ruled out. Okay, we are all a human being, right? Okay, who is the first human? We do not know. Okay, where did he come from? Where will he come from? Okay, now which generation we are living? All those things already we studied in the evolutionary aspects. Okay, so Homo sapiens erectus, Homo sapiens, Homo sapiens, Neanderthal. Okay, so likewise already we study each one. It has a specific properties. Okay, and functions also, brain size and habitats. Okay, all those things. Okay, see, this is the important. See that one, phylogenetic trees. This is the phylogenetic trees. See, this is a bacteria. Yeah, bacteria is also there. These are all considered, you know, about prokaryote, archaeobacteria. Compared to this one, same bacteria, but in this category, it can be grow in the marshy habitat. Okay, marshy habitat, methogen, methanogen product compound, thermoprotease. It can survive in the 96 degrees Celsius. Pyroticticum. So these are the halophilus in salt water. Likewise. And another one is eukaryota. From this one to this one. So there is a evolution aspects. See that one? Diplomata, Microsporidia, Entomoeba, Ciliata, plants, fungi, and animals. These are all considered eukaryotes, archaea, and bacteria. This is a phylogenetic disease, simple. Some of the plants or animals, some of the animals, particularly the microorganism. Take an example, plasmodium. Plasmodium, it contains number of microorganisms. Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, Plasmodium malaria. 4 is there. We know 4 only, isn't it? So many things is also there. See the next one, you can easily understand. See that one? Don't see the this one. Just imagine in this part. Here the Pteropus vampires, Pteripus alecta. Okay, these are all are same category. Myotius lucificus, Myotius davidi. So these two are the same. Likewise, how it will be related one? Okay, we are all are related to human being. Before that one, Homo sapiens is the last one. Before that, Homo habilis, Homo erectus. Okay, likewise. So first one origin is the Homo erectus. Okay, likewise. So how it will be related? How it will be the human beings are related? How many things is there? How it will be related? What are the characteristics? Each one it has a specific. What is that? Characteristic, related one, similarity, dissimilarities. Okay, they can identify and also put as a T-lex. Likewise, okay. So many things is it. Just to go and check it in your Google, you will get the ideas. Okay. The last one. What are the advantages? What are the advantages? It can produce a large amount of the identical DNA molecule from the minute amount of starting material. This is the important advantage. And the technique is extremely sensitive. Okay. The DNA need not be pure for amplification. No need of amplification for pure. It should not be pure in manner. It may be a impure also. It can be amplified. That is very, very important. And the next one is obtaining foreign DNA molecules. Obtaining foreign DNA molecules. Foreign DNA gene product. How it be? When you insert a piece of alien DNA into the cloning vector and transfer it into a bacterial plant or animal cell, the alien DNA gets multiplied. Already we discussed so many times, isn't it? Already we saw it, that things also. Suppose take an example. Okay, take an examples. See that? Yes. Just a minute, my dear. See that one? When you are inserting piece of alien DNA into the cloning vector and transfer into the bacterial, plant, animal cell, the alien DNA gets multiplied. Already we saw it, these things, isn't it? So no need to worry. See here. Already we saw it so many times. See, again, I will tell you. So this is considered a plant, isn't it? Same things is also repeated because it is here. So this is considered a plant, isn't it? Plant. The plant cell already know everybody, isn't it? Plant cell, it has a nucleus and the other cell bound organelles. So here there is a nucleus is there, isn't it? Nucleus. But the extra bound organelles called as what is that? This is considered a plasmids. Okay. So this is the plant cell. This is a plant cell, isn't it? So this is nucleus. Okay, nucleus. Okay, this one is called as plasmids. Okay. Okay, this one is called as plasmids. Okay, the plasmids comes out, it means the structure will be already I draw it, isn't it? So this is the sequence plasmid. Extra chromosomal body present inside the cell is a plasmids. Okay, we can isolate the particular desired gene. Already you know about the desired gene is okay. We are going to isolate. Okay, then only it will be better for understanding. So this is the desired gene. Imagine this is the desired gene. Any one of the resistant properties or any disease resistance or any other particular gene. It can be cut out. Okay, you will cut out means you will get it, the structure will be like this. The structure will be like this, isn't it? See this? Okay, this is the responsible, interested gene, interested target gene. Okay, isolated. So another one is how it will be. The remaining part will be how it will be like this. Okay, the remaining part will be okay. You will get it. This part and this part. Okay, this one we are going to what? Amplify. Okay, amplification means it can be entered into the another samples. Okay, it can be joined with the another samples. Aliens DNA molecule join here, so you will get the result. Okay, so both are together, joined together here. Okay, I will get it like this, isn't it? This is the interested gene. So after the completion, here there are so many enzymes also involved. The cutting process and the ligation process, all are the DNA polymerase cutting process. Ligation is also involved in. After the completion of these steps, it may be enter into the Dicea bacteria. That is called as maximum. The vector is E. coli bacteria they were used. It's also inserted. Okay, inserted. What happened inside? Inside there is a multiplication takes place. Inside there is a multiplication. These are the two combination of the two genes present in it. Two genes. Okay, the two genes inside. Okay, we are using electroporation and also what is that? Another one. What is that? Okay, this one. So now finally what happened? You will get so many multiplication copies inside. Okay, because inside we'll get the number of copies. That is very, very much similar to that of the two genes. Okay, so this is the process. This is the process. First step, once again, selecting the cell. The cell already know about. There is a plasmid is there. The plasmid is isolated here. Plasmid. After the plasmid isolations, you have to add the desired genes. You have to identify the desired genes. Okay, and the desired gene cut and out and separated by the technique. So many technique is there. Molecular scissor we were used. After that, join with the another DNA sequence. This one is called as B. This one is considered as A. Okay, so here the both the DNA will be present in A and B. So it inserted into the E. coli. This is the E. coli is nothing but the host. E. coli is the host. So finally, what happened now? The sequence is to this compound. Okay, you will get it millions of copies. That is very, very important. Millions of copies. Millions of copies you will get it. This is the technique. This is the technique they were used. So number of copies here. These are all what is that? These are all important one. This is the final step. Okay. Final result. That's very good. These are the things. Okay, my dear. Okay. So see here. So almost all recombinant technology, the ultimate aim is to produce a desirable protein. So which one is the end of the reaction? You know, desirable protein, not the DNA, not anything else. DNA is also very, very important, but particularly desired protein molecules, it will be very aim. Ultimate aim is very, very important one. So ultimate, which one is the produce the desirable protein? Which protein we need? That protein only we can produced by this technique only, and there is need for a recombinant DNA to be expressed. Okay, that is a very, very important. The foreign gene get expressed under the appropriate condition. The expression of the foreign gene is host cell involved in understanding many technical details. After having cloned the gene of the interest and having optimized the condition to induce the expression of the target protein, one has to consider producing on a large scale. The protein is not a single one. It is produced in the large scales, not only single one. Okay, not only single one. You can produce the number of copies. How much you know? Lot of copies we can produce. That is the important of this technique. The protein encoding gene expressed in the heterologous host from the recombinant. Heterologous means you know the different strains. Recombinant protein and self-harboring clones genes of interest may be grown on a small scale laboratory. Okay, we can create the single lab is enough for the creating the recombinant protein molecules. Okay, the culture may be used for the extraction of the desired protein and then purifying it by using the different separation technique. Different separation technique, you know about that. One of the technique is called as electrophoresis and also chromatography. Suppose if it is purification of the enzymes and hormones, the things what happen with the technique is involved. Which technique? Biochemical technique. Which biochemical technique? Chromatographic technique, chromatographic, and then paper chromatography, thin layer chromatography, TLC. Okay, thin layer HPLC, like that it comes. Okay, so we'll see the remaining things on the next class. We will finish up to the level. I will summarize all those things within short period. Okay, see that one. What are the things we discuss here now? See here. Yes, gross cell electrodes already saw it. Okay, see here the polymer exchange reaction already we saw it, and component of PCRs. Component of PCRs. Template is very essential one, and primer is also very essential. Okay, so after that introduction of the primer, what happened? There are two types of primers. One is called as forward primer, another one is called as a reverse primer is also there. So already you know about this is the forward primer. This one is called as a reverse primer. They may complement, which is the mother genes, isn't it? So what is the properties of primer? Already you know that primer should be in 18 to 25 bases in length, and also the melting temperature should be 60 to 65 degrees Celsius. And the primer should not be self-complementary. It is not complement itself. The content of the guanine and the cytosine is based on their how many percentage? 40 to 60% is there. It should be there. And then what are the deoxynucleotide means? There are 4 already in our what are the nitrogen bases present in it. That things also. Divalent cation, magnesium, and also Mn is very essential, particularly the Taq polymerase. Okay, thermal cycler. Thermal cycler is a instruments, otherwise called as machines used for the PCR process. Which principle they were used? Peltier effect. Peltier effect principle they were used for the PCR technique. Okay, so that is also a very important things. And then steps, first of all, melting, which temperature, which bases, how many times it will take, 100 to 500 base pairs should be there, and 94 degrees Celsius up to 20 to 30 seconds, and the melting of the target genes, and then denaturation and the melting process also done. So how many minutes it will take after that? You have to undergo the annealing process. Okay, 45 seconds for the about 30 to 40 seconds, it will be finished. Okay, so this is the structure, entire sequence, the entire process, how it will be occurs. All those things already we discussed very deeply. Here you must note it on 3 temperature is that. That is very important. 95, 55, and also less than. Okay, suppose we are adding the DNA molecule extra means the temperature should be in 70. Okay, below 70 like that. Okay, 68. Okay, don't forget. So here that there is 3 processes: the denaturation, annihilation, elongations. Okay, one by one we are seeing. So annealing process, how many temperature, how many minutes? Okay, what happened? That is very important. And then which enzyme is involved? Which enzyme? Polymerase is the enzymes used for the DNA strand. Okay, so then next one is after 20, how many cycles it will take? There are 3 steps are involved. After 20 cycles, approximately 1 million molecules are cloned from a single segment of double-stranded DNA molecules. That things also already we discussed. And then PCR reactions already know about how many steps is involved. Cycle 1, cycle 2, cycle 3, cycle 25. How much is that? And application of PCR is very important. Okay, where there is a use is an amplification of the DNA molecule, diagnosis of disease, anemia, penile kidney disease finding, and also forensic, criminal, and generation of probe, and the cDNA library, archaeological sample, and also RNA amplification. In paleontology and PCR in veterinary parasitology, phylogenetic analysis, and also which one and advantages. What are the advantages present in it? Okay, that things already we discussed. Thank you so much for the participation. We'll see the remaining things on the next class, my dear students. Thank you so much. Bye. Take care. Happy Pongal, all of you, my dear students. Thank you.
Thank you, sir.
Thank
Wish
you.
the
Thank you,
best.
sir. Happy Pongal, sir.
Thank you, ma'am. Thank you so much, ma'am. Thank you so much for your family, ma'am. Same wishes, ma'am.